year unfortunately had to cancel, but we were very lucky at the very last notice that Moustapha Kassen was able to step in and tell us a little bit about, I think, bone and skeletal aging, maybe?
Yeah, bone aging, stem cells. First of all, I would like to thank Martin for giving me this opportunity to talk to you, and also for you and Alex for creating this meeting. It's actually, I've been to many aging meetings, but this meeting is full of youthful energy, and it has actually rejuvenated me through these last 4 days. I'm going to talk, actually, I would like to thank Becca because he introduced some of the concepts related to stem cell. So I am a clinical endocrinologist, and in my talk, I actually will do the opposite of what you heard in other talks. So I do a reverse engineering approach. I will start with the disease that I have been interested in for most of my clinical and research life, which is osteoporosis. I will just get, yeah. So actually this photomicrograph illustrates what osteoporosis is. It's, as you can see, this is a cross-section of vertebra. So on your left-hand side, a vertebra taken from a young person, and on your right-hand side is a vertebra taken from an elderly person with osteoporosis. It's very clear what osteoporosis is. It's a disease of decreased bone mass and architectural deterioration, leading to increased bone fragility. So when the patients arrive to us, they arrive to us because of fractures. So fracture is a major healthcare problem. It affects both women, as you can see on the graphs on your left-hand side, and men on the graphs on the right-hand side. And actually, it is also age-related phenomenon. The bone field actually has been focusing for many years on sex steroids as the main cause of osteoporosis. So it was what's called the estrogen-centric view of osteoporotic pathophysiology. But during the last years, there is an increasing interest on studying what's happening inside the skeleton as we age, because as you can see, both in men and women, there is an increase in the incidence of fracture with age. Actually, for men also, this is a new recognition that they are affected by osteoporosis in spite of the fact that their incidence is delayed around 10 to 15 years compared to women. So just a quick review of one of the principal concepts in bone biology. You've heard about this intestinal regeneration, but actually bone, in spite of the fact that it's counterintuitive, it's a solid organ, but it undergoes continuous remodeling. So this continuous remodeling had the advantage of removing old bones full of what's called the microfractures, fatigue fractures, because it's exposed all over the time to mechanical stress, and replace it with a new bone of better biomechanical competencies. Actually, in anecdote, so just to remember it, while you are sitting here in front of me, there is around 1 million microscopic foci undergoing remodeling in your skeleton. If you'd like to help your skeleton, you need to jump a little bit, which is, we've been sitting quite a long time. Maybe we should jump a little bit to enhance our bone formation. But what's bone remodeling is actually a kind of a unique process because it starts by removing old bones by cells that are called osteoclasts. They are very related to macrophages. They contain bone-degrading enzymes, so they remove the old bones. And afterwards, when they finish, they call these bone-forming cells. They are coming from stem cells, adult stem cells, as we heard. It's one of the adult stem cell systems that we have in our body, as we heard from the last talk about intestinal stem cells. But these are bone marrow stem cells that are recruited to bone formation surfaces and undergoing bone formation. When we compare bone formation dynamics between young and old, we observe actually that one of the major pathophysiological mechanisms underlying the age-related bone loss and osteoporosis-related bone loss is impaired bone formation. So the cells, the bone-forming cells in our skeleton, they lose this capacity to form bone as we age. We'll come back to why this is happening. But just to give you a real world view of how these cells look like, so what we know now that bone-forming cells, as you can see here, they are beautiful. We call them osteoblasts, and they are forming bone matrix. I would like, yeah, here. They are recruited from stem cells that are present at the outer surface of blood vessels that are always associated with bone formation. So they are usually here. So they are located in critical place to be recruited to bone formation during bone remodeling. But I would like to draw your attention to another very important cells inside the bone marrow, which is the adipocyte, which they are here. So these stem cells, they are capable for also forming bone marrow adipocytes. And bone marrow adipocytes, this is a kind of a whole organ of adipocytes that are present inside the bone, and it plays a role in providing energy needed for hematopoiesis and for bone remodeling, which requires a lot of, or it's an energy rich process. So this ability of stem cells to change between these two fates, bone formation and adipocyte formation, is very interesting, because when we quantify the amount of adipocytes and the adipose tissue volume inside the bone as we age, we observe this phenomenon that adipogenesis increase inside the bone as we age. And this is associated with the very well-known process, which is age-related decrease in bone mass. So these two processes are linked. And actually, one direction in my lab deals with how these stem cells choose this differentiation lineage, either to osteoblasts and adipocytes. And this is a study performed by Alex Rauch in my department, who actually studied the gene expression of mesenchymal stem cells or skeletal stem cells as they go into osteoblastic phase or adipocyte phase during around 14 days of in vitro differentiation. And they characterize what happened at gene levels, and genetic pathways are induced either during osteoblast differentiation or adipocyte differentiation. And what's interesting in this study, that these stem cells, they are actually, their default differentiation pathway is into osteoblast. As you can see here in the promoter analysis and the chromatin analysis of several osteoblast-specific genes, the enhancers are already active at the stem cell stage. So they need only just a small push to differentiate into osteoblast, whereas adipocyte differentiation required more extensive chromatin remodeling. It's a paper rich in data, so if you are interested in this topic, you can go back to it. But actually what we observe, it's not only in osteoporosis where we observe this phenomena of increased adipogenesis, but also in condition what we call secondary osteoporosis. And in my lab, we have been also interested in what happens during obesity and type 2 diabetes and type 1 diabetes. And why that? Because there is a new recognition that these conditions are associated with increased fracture risk, actually up to 6-fold increase in fracture risk. So there is an issue of bone fragility also during these metabolic conditions. And also these conditions are associated with increased adipogenesis inside the bone marrow. So we conducted a number of studies that I'm going through them rather quickly just to give you a flavor where we stand. So these studies were performed by a postdoc that was in my lab, but she now has her own lab in Prague, Michaela Tencerova. And we started by seeing the effects on bone of high-fat diet-induced obesity. And as you can see here, there is a detrimental effect. So this is mice fed on high-fat diet, and they exhibit reduced bone mass, both trabecular bone mass and cortical bone mass, compared with normal diet-fed mice. And also this is associated with increased adipocyte infiltration inside the bone marrow. We extended these studies into humans, and these were young human males, and we compared what's happening inside their bone, both at the tissue level, but also at the cellular and molecular level, going from lean to overweight to obese. We've done extensive studies, but here I just show maybe the most important slide relevant to this talk, that if we take cells from obese persons and expose them to differentiation conditions in vitro, what we observe that the obese cells, they form more adipocytes than cells coming from lean persons. Dalia Ali, who is presenting a poster at this meeting, you can have a look at it, performed a similar experiment in mice that were fed with high-fat diet to induce obesity, but she combined it with OVX. And we heard yesterday about how this estrogen deficiency promotes senescence. And because actually most of the obesity studies in mice were performed in male mice, so we extended these studies into female mice. And as you can see, actually there is an interaction between estrogen deficiency and high-fat diet. So in this condition, we get enhanced and increased bone marrow adipogenesis, and at the same time, we get more pronounced bone loss. So there is an interaction between these two conditions, and I think Dalia, she can present a lot more mechanistic data why this is happening. But also here, also for the last part of these translational studies, we also saw what happens in mice bone regenerative capacity when they are when we induce either hyperinsulinemia and obesity by high-fat diet, or as in advanced type 2 diabetes, insulopenia, where we combined high-fat diet with streptozoosin treatment. And as you can see, so this is a model of bone regeneration where we create a defect in the tibia of the mice, and then we follow what happened to bone regeneration. So as you can see on the top graphs, that in these mice, they have impaired bone regeneration and the closure of this defect compared to mice on control mice. And also we have done a lot of cellular molecular studies showing that also there it's because of increased adipogenesis, inhibition of osteoblast, and actually we saw in all these three studies, high-fat diet induced obesity in male mice, obesity in human, high-fat diet induced obesity in female mice combined with OVX, and in these fracture studies, all of them, they have actually accelerated senescence phenotype inside their bone marrow. And this senescence phenotype is the cause of changing stem cell differentiation fate from osteoblasts to adipocytes. Is there a possibility to do something because of my background in clinical endocrinology? So the idea was to try to find the factors that may change this differentiation fate from adipocyte into osteoblast. How can we do that? I was very interested in secreted factor inside the bone marrow that can control this differentiation fate. And in Denmark, we have very active proteomic groups. So this is in collaboration with Matthias Mann, that you heard yesterday. We performed proteomic studies, and we identified a number of important secreted factors within the bone marrow. And of course, you get this long list of factors, so we needed to make some choices. I show you here one example. It's called KIA1199. It's a secreted factor that is known to play a role in extracellular matrix remodeling. It plays a role in cancer biology, cell migration, cell motility. It has also a role in the brain, in hearing. There is a human disease where there is a defect in this gene leads to deafness, but its role in bone is not known. And this is why we we focused on this factor. And actually, these experiments were performed by Chen Li. She is a postdoc in my lab, but she has her own lab in China right now. The first experiment was to try to study, is this factor expressed inside the bone microenvironment? And this is in situ hybridization, showing yes, it is expressed into the progenitor cells inside the bone marrow, and especially the cells surrounding bone marrow adipocytes. Also, the second question, which was very important for us from a translational point of view, we choose a factor that we can measure its level in the bone marrow microenvironment in bone marrow plasma. And as you can see here in these graphs, this is where we measure the level of this factor, and we put it against in the y-axis. This is the level of the factor, and on the x-axis, this is a marker or a clinical score related to increased fracture risk. So as you can see, the increase of fracture risk is associated with increased level of this factor inside the bone marrow. And also in the lower graph, you can see that there is increased level of this factor in the bone marrow of patients with osteoporosis. So we move from there to try to understand what the role of this factor in a tissue level in vivo, and also we performed some mechanistic studies. So here is just to show you, this is an overexpression study in mesenchymal stem cells on your right-hand side. So here the cells are overexpressing KIA 1199, and as you can see in the graph, so all these markers are related to adipogenesis. So this marker enhances adipogenesis in vitro, and when we blocked it down by siRNA, it inhibits adipogenesis. So we move from here into in vivo, so created KIA 1199 deficient mice, and as you can see here by extreme CT scanning, we showed that these mice contain fewer adipocytes inside their bone marrow. Both the number and the size of adipocytes was decreased, and actually the opposite phenotype was observed. So these mice, they have enhanced bone mass. Both the trabecular bone mass, as you can see here, and the cortical bone mass, as you can see here, compared to the wild type control. So I think because of time, I can't go into details about the mechanism, but I can take it during question part. But also here to show that these mice that are deficient of KIA 1199, they have improved actually bone regeneration following the same model of creating a tibial defect in mice. I think this is I give you a flavor where we stand in at least in my group related to using what insight that we have from aging to target and improve bone formation in the clinical conditions of osteoporosis. So here is the main point. Senescent bone marrow microenvironments leads to impaired bone formation and enhanced adipogenesis in osteoporosis, obesity, and type 2 diabetes. And targeting skeletal stem cells or bone stem cell niche is a possible option for regulating stem cell function and bone formation. And actually, this is what I learned also during this meeting. If we can place osteoporosis in the general context of biology of aging, already I have been inspired, but there are multiple targets that can be used in bone biology, and they can help in abolishing and treatment age-related bone loss. So thank you very much.
Thank you so much, Moustapha. Really fantastic talk. Any questions for Moustapha?
Great talk. The gene that you found is very interesting. Have you any idea, or do you like to test whether it can also prevent age-related yellow to red marrow formation and has an influence on HSC maintenance with age?
As you know, when we test this, we test the presence of adipocytes. So when they disappear, so there is a kind of compensatory increase in bone and also some improvement in in hematopoiesis. But actually we haven't looked at hematopoiesis because it's not hematopoietic lab, so to speak. So we focus on bone adipocyte, but it's a good point actually.
What is the source of KIA in the bone marrow microenvironment? And do you see any other effects in other organs when you knock it out?
Yeah, it's actually a great question. So I didn't have time to show that. So what we know that here I showed the effect on adipocyte differentiation. I showed the effects on bone differentiation. We have published that it increases mobilization of bone. This is the area of our research. Bone stem cells. So this is also a mechanism that can enhance recruitment of osteoblast progenitor cells into the bone formation surface. But actually there is another aspect of this factor that is fascinating, and it's related to its role in bone marrow adipogenesis, because it turned to be that bone marrow adipocytes actually they play a role in the whole body energy metabolism. So these mice, the knockout mice, they have increased insulin sensitivity and they are protected from the deleterious effects of high-fat diet, both on metabolism and on bone. So actually, there is a kind of a crosstalk between bone marrow adipocytes and the other energy-regulating organs. It's a very interesting area. Actually, from bone biology, we are amazed now. A lot of models inside the bone, and when you do some genetic manipulation inside the bone, you find amazing effects on other organs. So it's a very interesting area of research right now in bone.
Very nice, super fascinating. Thank you so much, Moustapha. So perhaps this